Hi, my name is Laura Krueger, and I'm a graduate student in the Department of Biological and Agriculture Engineering at Kansas State University. Today, I'll be discussing the beginning stages of my research project of investigating Marion Reservoir Cyanobacteria harmful algae blooms through integrated modeling and data science approaches. In Kansas, we are all heavily reliant on surface water reservoirs for water supply and recreational activities. However, the usage of Kansas reservoirs and their quality of water have been greatly impacted by the growth of cyanobacteria blooms. Under the right conditions, cyanobacteria blooms can release harmful toxins into the surrounding environments and pose health risks to fish, animals, and humans that are in contact with the water. There are many known drivers that have been linked to the increase in occurrence of harmful algae blooms, but some of these drivers are still unknown and can be site-specific. This research aims to better understand the environmental and ecological drivers of cyanobacteria in Marion Reservoir through site monitoring and integrated modeling approaches. This flowchart provides a general path of the project objectives. The monitoring and data collection includes taking water samples at 7 discrete, spatially distributed locations around Marion Reservoir and collection of continuous in-lake monitoring data with a multi-parameter sensor that is located near the dam. The collected monitoring data and supplemental environmental data sets will be utilized to inform modeling approaches, including a watershed model, a lake model, a cyanobacteria growth model, and data-driven models. Overall, through collaborations with project team members, this project will aim to integrate modeling approaches to characterize and provide a prediction framework for cyanobacteria harmful algae blooms. Currently, this project is still in the initial phases of data collection, site monitoring, and preliminary modeling setup. In the lower center of your screen, it displays the initial nutrient analysis data from the discrete water quality samples that have been collected over the past year. This plot includes the total nitrogen to total phosphorus ratio on the y-axis and collection date on the x-axis. The total N and total P are both measurements of the various forms of either nitrogen or phosphorus that is found in the water sample. The total N to total P ratio values are often used to describe which nutrient is limited in the system or which nutrient that is present in the least quantity that controls the growth of an organism. In the initial nutrient data collected, the total N to total P ratio remained fairly constant across sites up until the observed harmful algae bloom event that occurred in late May. Then it was followed by a decline in the ratio values, which may indicate that the reservoir becomes nitrogen limiting. Continued nutrient data collection will continue to provide insights into the nutrient availability and possible relationships to algal bloom growth. Initial in-lake monitoring data is collected from a multi-parameter zone that is housed in a buoy near the dam of Marion Reservoir. The zone is equipped with several sensors that collect data, taking readings continuously every 30 minutes. This graph shows the measurements of chlorophyll in blue and the total algae phycocyanin values in orange over the collection dates. Chlorophyll is a measurement of all the green pigment produced by algae and cyanobacteria. The phycocyanin value is an accessory pigment that is produced by the cyanobacteria and can provide an estimate of the cyanobacteria production. The pictures above the graph are a call out of a harmful algae bloom event that occurred in late May and early June of this year. Several observed spikes in the phycocyanin value, the orange-colored line, were observed, and these spikes correspond to the observed date of the harmful algae bloom event, as well as the monitoring data collected by the Kansas Department of Health and Environment that indicated a hazard advisory level of the high presence of cyanobacteria cells. In conclusion, the first year of data collection and site monitoring provides us with insights to conditions present at Marion Reservoir throughout the year and during harmful algae bloom events. Continuing data collection and monitoring will provide more robust data sets to inform modeling approaches. Next research steps will be to begin watershed and lake modeling development to inform the cyanobacteria growth model. Research will also continue by working with project collaborators to integrate data science modeling approaches and provide harmful algae bloom prediction framework. With hopes that this outcomes of this research will provide a more detailed understanding of the dynamics, the occurrence, and impacts of cyanobacteria blooms in reservoir systems. Thank you all for your time.